guys, this is Malinki. Welcome back to my channel Voice of Malinki. Today we will talk about HIV life cycle. And if you are new in my channel, please subscribe my channel. And if you like my video, please do like, comment and share my video. There are different stages of HIV life cycle: binding, fusion, reverse transcription, integration, replication, assembly and budding. There are mainly these 7 stages of HIV life cycle and we will talk about them one by one. In our last lecture, we have talked about the structure of HIV, and there I have mentioned that HIV contains RNA. HIV contains RNA and it has 2 important proteins. Those are reverse transcriptase and integrase. Okay. Now we will start the steps. So first step is the binding. So basically HIV binds to the receptors on the surface of CD4 cell. So basically CD4 cells are helper T cells or Th cells. Now HIV specifically binds these cells. Now HIV binds to the receptors on the surface of these cells. So suppose this is the CD4 cell and it has few receptors in the surface, and the HIV comes to bind this receptor. Okay, now what happens? This is the HIV, here it is the RNA, this is the envelope, and here it has 2 important proteins like reverse transcriptase and integrase. Now what happens? So next step is fusion. So the HIV envelope and the CD4 cell membrane fuse. This is the fusion and it allows HIV to enter CD4 cell. So basically the complete virus will not enter the cell, rather the RNA will enter the cell and few proteins will enter the CD4 cell like integrase and reverse transcriptase. Now what happens? The next step is reverse transcription. So inside the CD4 cells cytoplasm, HIV releases and uses reverse transcriptase, which is an HIV enzyme, to convert its RNA into DNA. So what happens? Since HIV has RNA genome, first of all it should be converted into DNA. So here is the HIV RNA which comes from the virus and it needs to be converted into HIV DNA. So the conversion of DNA from RNA, this process is known as reverse transcription. So this will happen here exactly, and in this reverse transcription the enzyme that helps here is reverse transcriptase, and this is the HIV enzyme. Okay, now the HIV DNA is there. So this DNA enters the nucleus and combines with the cell's DNA. So this is the cell's DNA. This is the cell's nucleus, and here this black one is the cell's DNA, host's DNA, and this red one is the HIV DNA. So this DNA, HIV DNA, will be integrated in this host cell's DNA. And who will help in this integration? So basically the integrase protein that is coming from the HIV will help in this integration. So this step is called integration. Viral integrase enzyme helps to insert the viral DNA into the host cell DNA. Okay, next step is the replication. So once integrated into the CD4 cell DNA, HIV begins to use the machinery of the CD4 to make HIV proteins. These proteins are the building blocks for more HIV. So the HIV DNA is now integrated in the host cell's DNA, and after that the DNA replication will happen and then transcription will happen. So again HIV RNA will be formed from the DNA, and the host cells machinery, host cells, different enzymes will help in this replication process, in this transcription process. And once the HIV RNA is made inside the nucleus, the RNA will exit the nucleus and HIV RNA will enter the cytoplasm, and here translation will take place, and again different HIV proteins will be made in the cytoplasm. Okay, so next step is the assembly. So new HIV proteins and RNA assemble into HIV. So here we have the HIV proteins, HIV RNA. Again they will assemble into the complete virus, and then budding step, this is the last step. So newly formed HIV pushes itself out of the host CD4 cell. So here is the assembled virus, this is the complete virus. So it will push itself and it will bud in this way and will exit the cell, and again the virus, HIV virus, will attack the nearby cells. In this way HIV will complete its life cycle. So this is all about today's lecture. I hope you liked the lecture. Thank you for watching my video.